This is the way life begins, a human sperm entering a human egg. These are scenes from a highly controversial film made by Dr. Daniela Petrucci of Bologna, Italy, accepted by some doctors, not by others. This is the fertilized egg, its cells dividing. In the United States, human embryos have been grown outside the womb for 6 days. Dr. Petrucci reported this embryo lived for 59 days. Life begun in a test tube. What is life? How is it formed? Is it God-given and sacred? What if man could remake man in any image he chose? Would he choose wisely, or would he choose Huxley's Brave New World? These are profound questions for today, because in the 21st century, man may have the most awesome power in history to design human life itself. You are hearing the heartbeat of a 9-week-old fetus in an artificial womb in the laboratory of Dr. Robert Goodlin at the Stanford Medical Center. Fetuses have been kept alive here following miscarriages for 24 hours. Someday it may be possible to save fetuses aborted at almost any stage of pregnancy and carry them through to birth. The aim here, which is being pursued on many fronts, is to study that deepest of all human mysteries, the mystery of life.
The 21st Century is brought to you by Union Carbide. The Discovery Company.
Today, in the second half of the 20th century, at some of our foremost research institutes and universities, the great intellectual thrust in science is in molecular biology, probing into the very core and essence of life. Some of our finest minds are engaged in it. It has been said that when the history of science in this century is written, the first half will have been the study of things, the automobile, airplane, rocket. The second half will be the study of living things, the cell, the chromosome, the gene. The results will be critical to us all in the 21st century. The mystery of life is incredibly complex. When life reproduces itself, cells divide, and the result is a new life that combines the traits of both father and mother. In copying itself, life relies on a submicroscopic structure called a gene. On the human level, the complexity is overwhelming. Each of us has about 50 trillion cells. Each of these cells has 46 chromosomes. Each chromosome is made up of more than 1,000 genes. Stored in these genes, like tape in a computer, is all of the information needed to direct the construction of a living human being. Inside the gene is the exact blueprint for every human characteristic, like skin color, expressed in a chemical code. The name of the molecule which contains the chemical code is as strange today as radar or jet planes were 34 years ago. It will be as familiar in the 21st century. Its name is deoxyribonucleic acid, or DNA. It has been said that the 21st century will be the DNA age, just as the 20th century was the nuclear, electronic, and space age. In the DNA molecule lie the final secrets of heredity. DNA's genetic messages are written in a 4-letter code, each letter being a specific chemical substance. When nature alters the code, it is called a mutation. Today, scientists are just beginning to read the genetic code. When they can write it, alter the code, man-made mutations will be possible, and our powers could be truly godlike. We could make men taller, healthier, smarter, or even choose color. Then we might make carbon copies of the design. If we had a sample of their tissue and could manipulate the genetic code, we might be able to produce an identical twin of Plato, Mozart, Einstein. Will this happen? Walter Sullivan, the science editor of the New York Times.
It's going to be a long, tough thing when you think of the enormous complexity of the genetic code as embodied in that one little human egg that you can barely see with a naked eye, and the sperm that you can't even see with a naked eye. That in that tiny little dot, twisted up in all those coils of DNA, is the word, the code, the instructions for the construction of a whole human being. And how do you reach into that little can of worms and hit the right thing to do what you want? It's a long way off, I think.
To get into this micro world of the gene, Te-Ping Lin of the University of California School of Medicine is using some new and marvelously miniaturized tools. He delicately injects minute amounts of protein into fertilized mouse eggs and then reimplants them in foster mothers where they continue to grow as normal embryos. If human eggs could be grown outside the body, Lin's cellular and subcellular engineering might permit us to manipulate these eggs, to inject specific materials to influence their genetic development. Another example of genetic manipulation on a primitive level today is being conducted by Saburo Nawa of the National Institute of Genetics in Japan. He is able to change the eye color of moths. He removes DNA from moths that have black eyes. Then he injects this DNA into the larva of a moth whose parents both had red eyes. This moth will be hatched with red eyes, but its offspring, in about 1 in 1,000 cases, will have black eyes. In these cases, he has switched genes from one living thing to another. If this could be done in humans, you could correct an abnormal gene that today causes a crippling illness. In Nawa's experiment, we see the black-eyed moth, which donated the DNA, the red-eyed moth, which received the injection in the larval state. Its offspring with black eyes, which was genetically switched, a man-made mutation. We spoke of someday making carbon copies of man. At Cornell, Dr. Frederick Stewart is making carbon copies of carrots. He isolates tiny cells from the body of a mature carrot, puts them into a solution which includes coconut milk. This somehow triggers the cells into behaving as if they were the original fertilized eggs, and they begin growing into new plants. Normal sexual reproduction has been bypassed. There is only one parent, the donor carrot, and the offspring are identical to it, not different as when the genes of two parents are combined. This also demonstrates that in every cell of an organism are all the genes needed to grow a new organism. Dr. Stewart declines to speculate about human applications, but his experiments do suggest that at some distant day, a single cell taken from the world's greatest genius might be grown into a second identical genius. At Oxford University in England, identical frogs are being grown, also bypassing sexual reproduction. These frogs have only one parent, a tadpole. The method is called nuclear transplantation. Zoologist J.B. Gurdon takes an unfertilized frog egg and with infinite patience and precision removes the nucleus with a tiny pipette. Then he transfers into the egg the nucleus of an intestine cell from a tadpole. This makes the egg act exactly as if it were fertilized. The frogs that result are identical and man-made. Their one parent, the tadpole which donated the intestine cell. We asked Dr. Gurdon about future applications for humans.
Doctor, some of the futurists have suggested that perhaps in the further progression of experiments like these, you fellows may eventually get to the place where you can tamper with the actual makeup of the gene so that eventually we'd get the kind of man we thought man ought to be.
Well, I've read reports in the press about the possibility of making duplicate humans, and these reports appear to have been based upon the kind of experiments I've been describing to you. One can see that if it were possible to do this same experiment in humans, what one would do, for example, is to take the nucleus of a human intestine cell, implant it into a human egg, and could then put that egg back into a female lady, and it would then possibly be born as a normal individual. Now, first of all, let's consider what immediate consequences this would have. For one thing, we would in this way be able to produce lots of identical humans. They would be as similar to each other as identical twins are to each other, as they're produced at the moment. And if this could be done, there would be no limit to the number of such individuals that could be produced. And furthermore, it would even be possible to take a certain amount of tissue from a living individual, which they wouldn't lack. It wouldn't matter taking a few cells from them, and to produce more individuals like them. I myself think that there are very substantial technical difficulties in achieving this situation.
This mystery of life before birth is being probed at Washington State University by biologist E.S.E. Hafez. His belief, conception can take place in one womb, pregnancy in another. Hafez removes tiny embryos from artificially inseminated goats, studies them to see if they are alive. Then he transplants them into a rabbit where they can live 4 days before being transferred to another goat. He also does this with sheep embryos, making it possible for a whole flock of prize sheep to be shipped across an ocean inside a single rabbit. Further in the future, Hafez sees us sending to the moon frozen embryos, there to be grown into cows, pigs, chickens, horses, or people. Hafez also sees the day when a woman who could not carry a baby to term could have the embryo implanted in another woman. When born, the child would be returned to his biological parents, his genetic inheritance unaffected. What makes some rats smart and some stupid, like this one, unable to find his supper in a maze? Scientists believe one of the factors may be the number of nerve cells they have in their brains. This rat, known as super rat, has no difficulty. At UCLA, microbiologist Stephen Zamenhof is making rats smarter by treatment before birth. Zamenhof injects large doses of a pituitary growth hormone into a rat, pregnant for 7 days. When her litter is born 14 days later, they have more brain power. This slide shows sections of the rat's brains. At the right, 25% more nerve cells in the brain of a treated rat. It also is 30% heavier. Says Dr. Zamenhof, we see no reason why this should not be applicable to humans, perhaps even before the dawn of the 21st century. Genetic experiments with animals are one thing, with people, quite another. How might the human race be improved? Genetic surgery, prenatal control, selective breeding, as in racehorses? Biologist James Bonner, California Institute of Technology.
It's clear that if any country on the face of our earth starts a large-scale program of selecting better people, all the other countries of the world have to get in the act too and join up, or else they'll soon be composed of obsolete old-fashioned people like us instead of the new super people. So the idea will spread. Professor H.J. Muller at Indiana University has talked about this subject a great deal. His proposal is that each baby, when it's born, must donate some of his sex cells, sperm or eggs, and these are put in a deep freeze and just kept. The person leads his life and dies, and after he's all dead and gone, so the heat of passion is taken out of the matter, a committee meets and studies his life.
Well, during his lifetime, then, he hasn't had any children.
He's been sterilized and hasn't had any children in the normal way. After he's dead and gone, the committee meets and reviews his life and asks, would we like to have some more people like him? If the answer is no, they take out his sex cells from the deep freeze and throw them away. But if the answer is yes, then they use them to fertilize eggs similarly selected on the basis of review and validation of the person's contributions during his lifetime. He just doesn't get to brazenly go out and propagate his own genes without assuring himself and everybody else that they're the best possible genes. Some people criticize this proposed program, on the basis of, well, how would you know what characteristics to select for? How would people ever agree? But I don't think that's going to be much of a problem initially anyway, because there are some characteristics on which everyone can agree. I think if we just say we want to select for intelligence of all kinds, people could agree on that. I would think that this mode of improvement of our race might very well be started within the next 100 years. Certainly not by the year 2000, but let's say in another 50 or 60 years after that. To a biologist, it's a sense-making proposal.
Harrison Brown, professor of geochemistry, also of Caltech.
Suppose that each one of us were to attempt to write down what is an ideal man or woman supposed to be like. What are the outstanding virtues we should attempt to breed into our population? You might say intelligence, but what kind of intelligence? You might say attractiveness, but what kind of attractiveness? We are very successful in breeding animals because we have very simple criteria. We can breed racehorses, and we only have one criterion: can it run fast? We can breed cows in terms of how much milk they give or how much meat they'll give per pound of corn you feed them. We can breed sheep in terms of how much wool they give or how much meat they give. But with human beings, what are your criteria going to be? Are they going to be running fast? Are they going to be men who look like Clark Gable? Or are they going to be men who are unusually intelligent? It is difficult for man to judge himself.
There is no controversy, however, about certain medical aspects of genetics, reducing infant mortality, for example. This child is a living example. She is healthy and well, but she would not have survived had she not been treated before she was born. They call this new medical specialty fetology. The idea? A fetus is a treatable patient, a dramatic departure from the old belief that a child was beyond human control until the moment of birth. This is an operating room at Cornell Medical Center, New York. Dr. John Queenan, a fetologist, is giving a blood transfusion to a baby still in the womb. He passes a slender needle through the mother's abdomen into the abdominal cavity of the unborn child. A major part of the child's blood supply is changed. Due to a genetic difference, the child's blood is at war with his mother's. They call it Rh incompatibility. In some cases, babies born with this condition have their entire blood supplies transfused after birth. Dr. Queenan treats only those babies he believes would not survive until birth. He saves about 40% of them. Another new genetic approach involves healthy children, ages 6 months to 6 years. This child is having his blood tested at the Albert Einstein Medical Center in New York to see if he suffers from any of 6 genetic defects that can be treated at the present time. This program, called Genetic Alert, is a new form of preventive medicine. Someday, Genetic Alert may be as routine as giving a baby his shots.
Yeah, how could I?
If laboratory tests find the blood deficient in specific proteins, which are the ultimate product of the genes, a genetic defect is indicated. The aim? Treat it now, when the child is well, before the genetic disease strikes. Dr. I. Herbert Scheinberg.
If we can detect a genetic illness in a child before the child suffers from the disease, we have the chance, in many instances, of preventing the disease from ever causing any suffering or, indeed, death. But nobody has ever been able to measure how many bad genes there are, which means how many genetic diseases there are in various populations of the world. We hope that as genetic alert expands, testing more people for more diseases, that we will begin to get some idea of what geneticists refer to as the genetic load, the unavoidable bad number of genes that generation carries on and passes on to the next generation.
The Donald Wineys of Warminster, Pennsylvania, have 3 children. All of them are healthy, but one, Christine, age 2, might have suffered from cataracts, cirrhosis of the liver, mental retardation, or died had her genetic disease not been diagnosed in time. It is called galactosemia. Through an abnormality in a DNA molecule, she lacks the enzyme needed to digest a milk sugar called galactose. The synthetic milk she drinks is free of this sugar, and she is thriving on it.
Go get your jacket first.
In the 21st century, couples may routinely receive genetic counseling to forewarn them of the possibility of such diseases. For the Donald Wineys, considering additional children, it is essential.
Well, the first thing
They confer with Doctors Lester Baker and William Melman of Philadelphia's Children's Hospital.
Well, my first question would be, what are the chances of having another galactosemia child, such as Christine? And then why Karen and Mark, our first 2 children, were normal? I mean, there's no signs of this in them. Can we hope to have another child without this problem?
The odds are 1 in 4 that the youngster could have the disease. I think, though, that in making up your decision, you also have to keep in mind the fact that with early diagnosis, you can get a child as well developed and as normally developed as Christine. And knowing now that you're carriers, we would watch your diet a little more closely during pregnancy, and then at birth we would get some cord blood, and Dr. Melman's lab would be able to tell us what that baby has. I mean, even when they're having ice cream...
Today, medicine can do nothing to correct the abnormal gene Christine Winey has. She is healthy because medicine can control the effects. Children with other genetic diseases are not so fortunate. We cannot yet reach into the DNA molecule and perform genetic surgery for those who might need it. But there may be a day, many doctors believe, when this might be possible.
Chrissy, put this where it's supposed to be. That's a girl. Very good. Okay, let me listen over here, too. And here. And how about the belly button? Show us the belly button.
The Wineys and their children only touch the surface in the DNA revolution. We must face the possibility that someday we may be able to change human heredity, change man himself. That will be the true and the awesome revolution. What sort of a revolution will it be? Man, said Friedrich Nietzsche, is a rope stretched between the animal and Superman, a rope over an abyss. Will it be a revolution cloaked in secrecy, a revolution subjected to intensive public debate? What should we be thinking about now? Lord Ritchie Calder of the University of Edinburgh.
But the real greatness and meaningfulness of mankind is its diversity. Not its conformity, it's its diversity. And therefore we're going to start simply blueprinting the future generations. We're going to have a very uninteresting world, I mean, apart from everything else. You know, there are thousands of people, energetic, brilliant people, all concentrating one aspect or another of this molecular biology and DNA thing. We've got these great centers of life sciences going on. And presently they're going to come through. And if it comes through under secrecy, under wraps, the way the atom bomb came through, then we're going to be faced with something dreadful. And this is frightening, because who is going to determine all this? You know, who are the wise men? We just have a generation produced in which everybody says what father says goes. But who's father?
the 21st Century was filmed and edited under the supervision and control of CBS News.